Delhi are honored to welcome colloquium, Deciphering Bioregulatory Mechanisms in Health and Disease using Omics approach. Day 1 the before lunch to mark the closing of the day, followed by dinner would be held up. So to begin, I would call and I'll request our PhD student, our senior faculty, to start with Ganesh Stuti.
Suryakoti Samaprabha Nirvignam Guru Me Deva Sarvakaryeshu Sarvada Vande Mataram Sujalam Sufalam Malayaj Shitalam Sasya Shamalam Mataram Vande Mataram Shubhra Jyotsna Pulakitayami Pulla Kusumita Dhrumadala Shobha
There is some break in the voice. Is it from university side? The internet is not getting connected properly.
To begin with the first session of the colloquium, we would like to invite Professor Daman Saluja and Dr. Manoj Kashyap to chair the scientific session. Dr. Daman Saluja is currently a professor and director at Medical Biotechnology Laboratory, Dr. Bhim Rao Ambedkar Center for Biomedical Research, University of Delhi. Professor Saluja works on medical biotechnology with a chief committed transmitted diseases. Professor Saluja is a recipient of prestigious INSA Young Scientist Medal and LSS Memorial Award by India. Dr. Manoj Kashyap is an associate professor at the Amity Stem Cell Institute, Amity University, Haryana. His research interests include finding the
I can't hear you. Is it some problem on my end or?
There is a break in voice.
Yes, there is.
There is a lot of breakage in the voice. Is it a problem from the side or?
No, ma'am, there is some breakage in the noise. There is some problem. Maybe a
बस I
technical
was
glitch.
worried कि मुझे मेरे यहाँ problem है या
No, ma'am. No, ma'am. It's there. कुछ है. Maybe sometime they'll
I can
catch up.
start. There is no problem if everybody can hear.
ठीक है.
Very good morning, everybody. I hope all of you can hear me.
Yes, ma'am.
All right, great. It's my pleasure to chair this session on deciphering bioregulatory mechanism in health and disease using Omics approach. The session first of today and starting of the conference or the colloquium. I'm really happy to welcome Dr. Dipali Sharma, who is Professor of Oncology currently at Johns Hopkins School of Medicine, USA. She is affiliated to Women Malignant, to the Institute working in the group of Women Malignancies Disease Group, Department of Oncology, Sydney Kimmel Comprehensive Cancer Center. Dr. Dipali Sharma has an interest in breast cancer, and they are looking from all angles, not only just single basic studying, trying to studying the signal mechanism and all, metastasis and stuff, but they are looking from all angles, metabolomics, microbiome, novel therapeutic strategies, therapy resistance, obesity, and its correlation with the cancer. So they are looking at the holistic approach from all angles, trying to elucidate the molecular mechanism underlying breast cancer initiation, progression, and developing various preventing and treatment strategies using initially as a mouse model and then going to the human samples. Her area of specific interest includes understanding the molecular connections between breast cancer and obesity, racial disparities, and microbial dysbiosis. Dr. Sharma's lab has uncovered that a pro-oncogenic colon microbe potentiates breast cancer initiation and progression. Her current projects aim to understand the mechanistic link between microbiota and breast cancer and develop strategies to harness bugs to modulate the response to drugs. Little bit about her biodata just to motivate the youngsters. She's BSc and MSc with specialization in endocrinology and biological chemistry. She was awarded her PhD degree in molecular biology oncology by Delhi University. She did then her postdoctoral fellowship at lab of Dr. Joseph Fondel at the University of Maryland. Hello.
Ma'am, you're audible. Ma'am, please
Yeah,
continue.
everybody else is able to hear me.
Yes, ma'am.
Okay. So sorry about this interruption. So then she joined Dr. Nancy Davidson at Johns Hopkins School of Medicine, and then she joined Winship Cancer Institute, Emory University as an assistant professor. And then, of course, currently she is at Johns Hopkins School of Medicine. She will be, today she will be enlightening us about her area, unraveling the mechanistic connections between microbiome and breast cancer, an ever-evolving story. And this is now, these days, as all of you know, microbiome and gut microbe is like the talk of the world, global talk. And we are associating every disease with change in the gut microbe and trying to, and I'm sure we will have it will be a very, very interesting talk. I'm looking forward to it. Dr. Dipali Sharma.
Thank you so much, Dr. Saluja. Thank you so much. And I would like to thank the organizers for giving me this opportunity to come and share some of our research with everyone here. And as you might have noticed, University of Delhi is my home, and I was at Department of Zoology for my master's and part of my PhD also, and then I moved to JNU. So this is, practically coming home. So I'm very happy to be here with all of you today. I hope you all can hear me fine.
Yes, Dr. Dipali, we can hear you very well.
All right. And am I allowed to share my screen now?
Please go ahead. I hope we will have no issue in that.
All right. I think since someone from the organizers is sharing, so I can't share.
So they have.
So I think they have to stop sharing.
Now,
now maybe I'll try now.
Now you can share.
And then let me know if you see it okay.
It's such a gratifying, feeling when your own alumnus come and give a talk in your own university. It's really a proud moment for Delhi University also.
Thank you so much. It's great. It's just great to be here with all of you. Can you see my
yes,
full
we can
screen,
see your full screen. We can see your screen and everything is clear,
okay,
and
great.
we can hear you.
Yes.
Okay, very good.
Okay,
Thank you so much. So I'll I'll start my talk by just telling you a little bit about my lab. So as Dr. Saluja mentioned, we have been looking at breast cancer and various aspects of breast cancer initiation and progression. And today I'll share some of our stories that are upcoming with microbiome and metabolome and genomic features of breast cancer. And first and foremost, I always try like to acknowledge the people behind the stories beforehand. So these are some of my pictures from some of the people from my lab. This is Hopkins, the famous dome building, and this is my lab during the holiday season. We were all dressed up in red and green mostly. And some of my collaborators, and of course, funding from various institutes, without which, of course, we can't do anything. So let's start this discussion about breast cancer and think about prevalence of breast cancer worldwide. So as you can see here, breast cancer, pink denotes breast cancer. And so the prevalence of breast cancer is truly worldwide. And you can see that how this is the most common cancer among females. And it is pretty much all over the world. And if you look at the incidence of breast cancer, it is disproportionately high burden in North America and some other European countries. And of course, here is India, where the burden is not that high. But if you look at the mortality is disproportionately high in African countries. And here, and the reason can be socioeconomic reasons, maybe some genetic determinants, maybe the prevalence of some of the aggressive forms of breast cancer, one of which is triple negative breast cancer. So maybe because of that, the burden of mortality is higher in these countries. So if you look at back at, as we pay more attention to the US breast cancer statistics, so we can see that how besides skin cancer, breast cancer is most commonly diagnosed cancer among American women. And about 30% of newly diagnosed cancer in women will be breast cancer. And about 1 in 8 US women, and this is about 13%, will develop invasive breast cancer over the course of our lifetime. And if you look at the new cases of invasive breast cancer, about 50,000 women will actually develop some new cases of cancer in situ. And if you look at invasive breast cancer, about 2,700 new invasive breast cancer cases are expected to be diagnosed in 2022. And about 43,000 women will succumb to the mortality, breast cancer related mortality. With all these grim stats, and I have to apologize now that I don't have the stats for India, but with this grim stat, we still have this good news to share that we have about 3.8 million breast cancer survivors in US alone. And of course, the numbers are higher around the world. And we can easily attribute these survivors to early diagnosis and development of novel therapeutics. As we understand the disease more and more, as we are developing novel therapeutics, the number of survivors is increasing. And of course, the number of women who are succumbing to this disease is continuously decreasing. So if you look at the histopathological classification, and this slide, I always try to include these slides for the benefit of students, so we can appreciate how breast cancer develops. So the cancer can develop in lobules. So these are lined up by epithelial cells, and these epithelial cells can proliferate and fill up the lobules. And once it is contained within the basement membrane, it is called lobular carcinoma in situ. And if the cancer develops here in the ducts, then it's called ductal carcinoma in situ. Eventually, these cells that are filling up the ducts by their uncontrolled proliferation, they will learn how to invade out. And this is, we can call a cancer cell as a rogue cell, which is going to basically win over all the obstacles that are put there, because all the cells in our body they know when to divide and when to stop dividing. But these cancer cells will evade all the checkpoints and they will divide uncontrollably. And these cells will eventually learn how to migrate through this and invade through these basement membranes that are basically containing these cells. And 5% of all breast cancers are invasive lobular carcinoma because they're generating in lobules and then they are learning to invade out. And similarly, invasive ductal carcinoma is when the cells are filling up the ducts and then they're learning how to invade out and become metastatic disease. And about 78% of all breast cancers are actually invasive ductal carcinoma. So it's pretty predominant. If you look at the risk factors that are associated with breast cancer, as we discussed earlier, about 1 in 8 women are at the risk of developing breast cancer in her lifetime. And of course, we have come a long way, as we discussed earlier. There are several modifiable and non-modifiable risk factors, and we have a lot of risk calculations that are available so that we can predict someone's risk for breast cancer. So some of the risk factors that include genetics, family history, genetic testing. Some of them are lifestyle related, and then environment related, toxic exposures, radiation, asbestos exposure, and also some of the personal history. Dense breast, pre-malignant disease. Some people have HRT or they have taken HRT or chronic inflammatory disease, and of course race and ethnicity. They're also a important part of calculating someone's risk. Now, several risk prediction models are also available and they are in use actively. But an intriguing aspect is that despite all these advances in prediction modeling and understanding cancer, about 70% of breast cancer develops in women who are at low risk. And actually, some of them have no risk of developing this disease. And that brings us to a point where we are forced to think beyond the usual suspects and incorporate newer aspects, such as microbiome. So now, some of us, like as Dr. Saluja was mentioning, that we are actually thinking a lot about microbiome these days and associating the changes in gut microbiota to various diseases. So that would mean that many of us in the audience actually do appreciate that trillions of bacteria live in our body and they constitute the microbiome. And as you might know, that Human Microbiome Project that characterized the microbiota of 5 body sites, including mouth, nose, colon, vagina, and skin. And this was a project that was funded by NIH, National Institute of Health. And for many days, so after this project was finished, we all appreciated the impact of microbiota on various diseases, but various other organs in the body were considered sterile. It was in the past few years, microbiome field has grown so much at a surprising rate that now we know that there is a unique and distinct microbial communities that they exist in so-called sterile organs. Let's think about pancreas or prostate, and of course, breast. They were considered sterile, but now we know there is rich microbiota that lives in breast. So I would say that a couple of years ago, say in 2014, as we were kind of growing to understand breast microbiota, this is the study that I wanted to share with you that shows the presence of breast microbiota. And so this study used paired normal, adjacent tissue, and tumor tissue of 20 patients. Those were all estrogen receptor positive breast cancer. And then the sequencing showed that how 5 richest phyla consisting of proteobacteria, as you can see here, firmicutes, actinobacteria, bacteroidetes, and verrucomicrobia, that accounted for the majority of microbiome in these particular samples. And as you can see here at the lower end, the bacterium Spingomonas was the most significantly enriched and prevalent in paired normal, which is the top panel here, and the bacterium Methylobacterium radiotolerans. That was the most significantly enriched and most prevalent bacteria in the tumor tissue. So that showed that not only breast contained microbiota, this already has some changes that were noticeable in paired normal and tumor tissues. And then this study actually tells you that how when these breasts were sampled at different places. So this is a unique design where samples were collected at multiple sites all around the breast. Because as I shared the structure earlier, there are various ducts and lobules in breast tissue. So in here, samples were collected from multiple sites all around the breast, and blue ovals represent the location of the tumor, and purple squares represent the location of the specimen obtained for the bacterial analysis. Now, several interesting observations were noted here. And as we're going to see this kind of microbiome data a lot during my talk, each bar represents a subject, and each colored box is a bacterial taxon or a phyla or a species, whatever that I'm discussing in that data. And the height of the colored box represents a relative abundance of that organism within the sample. So the most abundant taxa, and so this study actually has 2 sets of samples, one from Canadian and then one is from the Irish cohort. So the most abundant taxa in the Canadian sample was Bacillus and Acinetobacter. And in the Irish samples, the most abundant taxa was Enterobacteriaceae and the Staphylococcus. So there were, again, very distinct changes among the cohorts. Now, multiple studies over the years, they have analyzed nipple tissue aspirates, fine needle aspirations, and surgical samples, and they have shown the presence of breast microbiota. Now, this is a recent study that showed the load of bacterial DNA that, it was actually highest. So this is actually very interesting that over the years, as we can appreciate, since this is a symposium looking for, the multi-omic analysis, we can appreciate that how over the years we have come so far that once an organ breast was considered sterile, nobody believed that there was bacteria there. But now we appreciate that how, if you compare all these other organs, breast actually has the most abundant load of bacteria. In fact, the highest was present in breast tumor tissue in comparison to multiple other cancer tissues that were involved here. And the IHC analysis actually of the tumor tissue showed, now this IHC analysis used antibodies against bacterial lipopolysaccharides, LPS, and then FISH was used using a universal probe against bacterial 16S ribosomal RNA. And that showed the presence of bacteria in breast cancer tissue. Another interesting aspect is that breast tumor microbiota is actually richer and it is more diverse than all other tumor types. So these are all the box plots here, and these boxes are basically showing you Shannon diversity indexes for all the tumor types. And these boxes below here are showing the numbers of bacterial species present in each tumor. So about 40 samples were included in each tumor type, and we can appreciate that how the richness is higher in breast tissue as compared to all the other tissues. So it is interesting to note that how microbiome can interact with various hallmarks of cancer. So here, I think the audience members in audience would be familiar with cancer hallmarks, and as you might know that they have been revised also after the first initial paper. And so many of the microbiome members, these microbiota, they can actually interact with various hallmarks directly or indirectly. So some microbes actually would interact directly, and some of them will interact with via their microbial toxins, and they can directly alter all these processes, be it sustained proliferative signaling or tumor promoting inflammation, or genome instability and mutations. So if we focus our attention back to the established risk factors that I discussed earlier, and the factors that are related to breast cancer, and as I was discussing earlier, that we have been looking at breast cancer initiation progression from various angles. So if you look at these risk factors, microbiome also interacts with various other risk factors, such as estrogen, alcohol, breast density, aging, and obesity. So we can, so these are few of the risk factors that are actually very, very interesting. And if we now focus at, say, for example, estrogen levels. So breast cancer is associated with early age at menarche and late menopause, accounting for the overall exposure of a woman, overall exposure of to estrogen. So, and about 70% of breast cancer, you might remember I mentioned that 70% of breast cancer are estrogen receptor positive breast cancers, which are obviously fueled by estrogen. So now it is known that how microbiota is actually a critical regulator of estrogens in the body. Enteric bacteria have enzymatic activity and they're capable of deconjugating estrogens metabolites. And these deconjugated metabolites will actually, they will deconjugate these estrogens. And now these estrogen metabolites will actually, they will be fueled back, they will be reintroduced into circulation, and that would increase the systemic level of estrogen. And of course, there is enhanced bioavailability of androgens and progesterones, fueling again the complete estrogen levels. And of course, there is synthesis of estrogen mimics from dietary polyphenols. So with all these processes, gut microbiome can actually elevate someone's exposure to estrogen. Another very interesting risk factor is, I would say, alcohol. And alcohol is also associated with 7% increase in risk of developing breast cancer with moderate intake. And alcohol is also associated with gut dysbiosis. Now, one more risk factor, which I would say that not associated with every cancer, but it is directly associated with breast cancer, because breast cancer is an age-related disease. So age is another important risk factor. About 80% or so breast cancers would develop in women that are about over 50 or 60 years of age. And of course, the probability of developing breast cancer increases with age. So as we can appreciate from this cartoon here that I'm sharing, microbiome actually changes with age, and this cancer aging relationship as with the human microbiome is very dynamic, and it continuously evolves through the age and of course exposure to the environmental factors. High breast density is another risk factor associated with breast cancer, and breast, as we discussed earlier, it contains lobules, ducts, and fibrous tissue, fat, and of course, extent to fibrous and glandular tissue determines the breast density. So when women were characterized into low versus high breast density and their gut microbiota was analyzed, it shows that how firmicutes to bacteroids ratio that changes, and firmicutes to bacteroid ratio and microbiota diversity was greater in women with high breast density. So it's very interesting to note that how all these risk factors can be modulated by microbiota. So now obese state is also associated with microbial dysbiosis, and that contributes to dysregulated energy harvest and energetics. So enhanced energy harvest efficiency of an obese microbiota that can be a result of multiple processes. For example, if you can focus here, this is lean with healthy microbiota, and this is obese woman with dysbiosis. And in lean person, microbial fermentation of indigestible polysaccharides is lower, and this obviously opposite in obese state. And absorption of monosaccharides and production of short chain fatty acids is also lower in lean, and it's higher in the obese state. And also, if you look at the gut microbiota, there is a change in ratio. As you can see, formicutes are lower and bacteroides are actually higher. As this changes with obesity, and formicutes to bacteroides ratio actually changes. And my lab has been actually working a lot, looking at the mechanistic links between obesity and breast cancer, and we've been focusing on adipocytokine dysregulation and low-grade inflammation that actually does impact breast cancer initiation. So as I said, that our group has been examining links between obesity and intentional weight loss also. So with intentional weight loss, I mean caloric restriction or some other dietary modulators to achieve that healthy BMI and healthy microbiome in breast cancer patients. So this is a little piece of data that I'm sharing here. This is from a clinical study titled QUINE. And QUINE was an acronym for Cancer, Obesity, Overweight, and Insomnia Project. So in this project, we used a weight loss intervention, POWER, and POWER is for practice-based opportunities for weight reduction. And these participants were all breast cancer survivors with high BMI. So they all underwent active POWER remote intervention. And approximately, I would say 50% of participants, they did achieve weight loss. And their stool samples were collected at the baseline and at the completion. And interestingly, we observed differential abundance of genus here. And we found that there was a differential abundance of genus Bifidobacterium and Akkermansia in responders only. And it's important to note that how Bifidobacterium species are probiotics, and Akkermansia is a genus in the phylum Verrucomicrobia, and Akkermansia are also beneficial bacteria. So it's very interesting to note that how these microbes change with interventions. And again, in the 6 months post-intervention samples, differential abundance of genus Clostridium and Doria were observed. And Clostridium and Doria, they're associated with promotion of obesity. So in obese women who actually did not lose weight, there was an abundance of Clostridium and Doria. And then several other clinical trials related to obesity and breast cancer are underway right now in our breast cancer group, and we're moving ahead with that. Race is another important modifier, but it's still not considered in the risk prediction models. As is visible here, we can see that how incidence of breast cancer is highest among white women, followed by Black women. As you can see these lines here, white is red and then Black is blue here. So we can appreciate that how this is blue line is here and white is up here. But if you look at the death rates are here denoted here, and the mortality with breast cancer is actually highest in Black women, followed by the whites and Asian. So what we did was we looked at breast cancer samples from different races. And okay, so we did metagenomic analysis from the TCGA data, and then we analyzed and compared the bacterial community composition of about 1,000 or so primary tumors. And we found out that there was distinct changes in the microbiota if you compare races, white, Black, and Asians. So everybody had different microbiome compositions in breast. And then we wanted to identify if there are any potential microbial biomarkers among different races. And we use this linear discriminant analysis. And here in the top left, you can see the level of order. Only two we were able to distinguish only 2 orders in breast tumors from white women. But here at the family level, we could identify few for every race. And then of course, here you can see that we could identify some more changes that were apparent in Black, white, and Asian women. So we can conclude here that race is also associated with changes in breast microbiota. Now, while the clinical studies were undergoing and we have multiple plans, we did some meta-analysis of all the existing data. And we found that how, if we look at all the patient data that is available for all the microbiome studies so far, we wanted to look for the common trends among these, different omic analysis. And what we found was, this data is comparing microbiota from paired breast cancer and normal tissues. And top graph shows the phylum distribution of local breast microbiota in cancer and nearby normal tissue. And bottom left is showing you mean percentages of major phyla in cancer and nearby normal tissue. And of course, here you can see the percentage of 3 major phyla: bacteroids, firmicutes, and proteobacteria. And here it shows the frequency of occurrence of abundant taxa in breast cancer in nearby novel tissue. So this is one more comparative analysis that I wanted to share before moving on. So this data compares microbiota from benign and malignant disease. So here it shows phylum distribution in local breast microbiota. And here it shows the mean percentages of, again, major phyla, as well as the percentage of 3 major phyla: firmicutes, bacteroids, and proteobacteria. And here, again, we are sharing abundant taxa among these 2 groups. So with our meta-analysis of clinical data, we examined microbiota of the breast, and then we did some metabolomic analysis to look for bacteria to the toxin connections. And what we found was that Bacteria fragilis is consistently detected in all the breast tissue samples, from benign to malignant breast cancer samples, as well as nipple aspirates. And in the metabolomic analysis, we found that how this bacteria actually secretes a toxin, Bacteria fragilis toxin. We call it BFT. And then we asked this question: this is actually a colon microbe, and what is it doing in breast? And does it actually have a functional relevance? So with that, we started our study looking at the impact of B. fragilis. Does it actually contribute to breast cancer? And for that, what we did was we examined the impact of ductal dysbiosis on normal mammary tissue. So for that, we used an intraductal approach, and we introduced the bacteria through the ducts, and then we observed for the presence of B. fragilis. So this is ETBF, which is enterotoxic Bacteria fragilis, short form ETBF, and this is the mutant bug, which does not produce the toxin. And what we found was that when we administer by intraductal administration, we can actually see active B. fragilis in the breast tissue, so it can survive there. And then we also observed the presence of the toxin here in the breast tissue. And as you can see here, there is, in the ETBF group, there is a lot of filling of these ducts, and there is, in the tripalm staining, shows that there is a lot of fibrous tissue. And PCNA, Ki67, pancreatin staining, you can see that all of these, the proliferative cells are actually abundantly present in the ETBF group. And this is a quantitative representation of the data. And then next, we wanted to ask, with enteric B. fragilis infection, would that impact breast epithelial cells? As we can see here, when the mice were actually infected with enteric B. fragilis, that it does impact breast epithelial cells, because we found these are the end of the terminal ends. And as we can see here, that these terminal buds were actually swollen here, and we could actually count them. And again, we saw that active B. fragilis was present only in the ETBF group, but not in the sham control. And we found that the significantly enlarged terminal end buds were more prominent, with all these bifurcations that indicated a proliferative and inflammatory response. And we also observed that how enteric B. fragilis infection triggers a hypoplasia-like features. So I'm just sharing this cartoon to show that how breast ducts change as the cancer develops, and how ETBF gut colonized mice showed prominent change. So this is with the ETBF colonization, ducts are getting filled up, and they are actually pretty much resembling a DCIS-like situation. And again, these cells are very proliferative, as is indicated by these IHC stainings with Ki67 and pancreatin. And also they show profound changes within the immune cell infiltration, as it is shown by the CD3 staining. And we also observed that how when we have enteric B. fragilis infection, that increases breast tumorigenesis and metastasis. As when these mice, this was a memory tumorigenesis, spontaneous memory tumorigenesis model. And if we infect these mice with B. fragilis, we can see that how this red line denotes tumor progression, and this tumor progression was actually higher. And this is the tumor incidence. Of course, tumor incidence was at a lower age, and the tumor multiplicity was also higher. And this is showing you the survival, tumor-free survival. And of course, with ETBF, they were getting tumors early on, and the tumor load was actually very high. And then we asked a question, what is happening with this toxin? So we exposed breast cancer cells to toxin, and I just want to look, just want you guys to look at this control and then 72 hours exposure of the toxin. And you can appreciate how epithelial cells, very cuboidal looking epithelial cells, are now changing into very mesenchymal phenotype in every case. So we also observed that how when you expose these cells to toxin, there was no change in the colony formation of these cells. But when we look closer, as you can appreciate here, this is the colony made in the absence of the toxin. But when you expose these colonies, cancer cell colonies to toxin, there's all these cells that are proliferating out. So these are all migratory cells. So we took these cells and put them to RNA-seq. And with RNA-seq, we could see that there is actually an induction of motility network with these. So we found with the microbiome analysis, we found the microbe, and then with the metabolomics, we could find the toxin that is secreted by this microbe. And now we are looking at all these gene network changes. And we found that these cells, these cancer cells that are exposed to this toxin, are actually, they have distinct changes in genomic features, and we are seeing an abundance of cell movement associated genes. And we could then look at the phenotype, and we could see that if you expose these cells to toxin, and they definitely do migrate faster. And this is a scratch migration assay. When you expose these cells to toxin, they actually migrate faster, as you can see here in the graph. And I do have a video, I think, later on. Yeah, here. So this is a microcapillary assay where we are looking at single cell migration. So these are all the microcapillaries, and these are all the cells, just little movies to show that how these cells are actually migrating through the capillaries. So when they're exposed to the toxin, they actually do migrate at a faster velocity and speed. And there's also an enrichment of stemness signature that we observed in these PFT treated cells. And this is a network showing the stem cell pluripotency pathway. And we put these cells into mice, and then we found that how BFT exposed cells were actually making multifocal tumors, and they were very aggressive in nature. And then we took these primary xenografts. So we exposed the cells to BFT, put them as primary xenografts, took these cells out, and take these tumor dissociated cells, put them in mice again, but this time as a limiting dilution assay. And in this assay, we can see that how tumor volume is, of course, higher in the BFT group. And of course, tumor-free survival is actually shorter. And this is very interesting that these cells have increased stemness. And as you might know that increased stemness would actually can impart secondary tumors, poor metastatic progression, and also poor response to therapy. And we took these now secondary tumors and put them to RNA-seq again, because we wanted to know that how these bacteria that we identified with this microbiome analysis, how that impacts the genomic features of these tumors. So we found that there was an increase in stemness markers, definitely. And we could see that in a physiological assay, the mammosphere formation assay. And here we could see that BFT exposed cells are actually forming higher mammospheres. And there is an abundance of ONS factors, OCT4, NANOG, and SOX2. And this is the ICC cytochemical analysis, just staining, showing the same. And this is Western blotting, showing the increase in stemness markers in these cells. And again, this is the pinwheel showing us that how BFT treated cells that were exposed to the toxin only for 72 hours are retaining this memory after approximately 3 months. And we could see that these tumor dissociated cells are still showing a higher invasion as well as lower adhesion. And if you remember the cartoon that I shared earlier, this invasion and migration potential is absolutely necessary for metastatic progression. And again, they retain the memory for migration, and this is showing us the transwell migration. And also these are tumor spheroids. And when these cells are grown as tumor spheroids, we are seeing again the higher migration from these spheroids. And then we also looked at these enrichment of any pathways, because again, now we are moving on from the genomic features and we want to look at the pathways, the signaling pathways. And we found that these tumors that were from the BFT group had an enrichment of Wnt, catenin, and Notch pathways. And then we went on and actually, and I'll explain this cartoon a little bit. So here I'm showing you the Wnt, catenin pathway. So this is, if this is our BFT, then it can actually, if it can impact beta-catenin, which can be in the dissociation complex. When the beta-catenin is in the dissociation complex, it is inactive. But if this dissociation complex is disintegrated, then beta-catenin is now free, and this will undergo nuclear translocation. So here we show that, of course, with the toxin treatment, BFT treatment, beta-catenin is increased at the transcription level as well as the protein level. And of course, there is more nuclear retention of beta-catenin as compared to the cytoplasmic localization. And it's also visible in the ICC analysis here. And we looked at all the downstream transcription factors to show that this BFT is increasing the expression of all the responsive genes. And similarly, we went on and checked the other pathway. And I'm going to, in the interest of time, I'm just going to be a little bit, I'm going to probably skip some data here. And we show that
you can wind up into couple of minutes.
Yeah, I think so, because I just looked at the clock and I'm like, I'm running out of time. Okay, so I'm just gonna show you that we did look at these pathways, and then this is showing you migration and invasion potential. And this is, we use these inhibitors to inhibit the actions of Notch and catenin. And we could see that how with the inhibitors, we could inhibit the impact of this toxin. And I'm going to just show you this here, that these are the two pathways that we found, the Notch pathway and the beta-catenin pathway. And we could actually inhibit these two pathways and inhibit BFT's impact. And I'm going to skip these metastatic data, and I think come here, because this is, I think, the most important slide of all, which actually tells you how to, there are so many opportunities to harness microbiota changes. And with all these omic analysis, now we have understood that there is distinct microbiota that is present. This microbiota can change the metabolome. And with this, all these metabolites can now go on and snowball these molecular features. And these molecular features, when they are attained by a tumor cells, like in this particular case, 48 hours exposure to bacteria or its metabolite can actually change the molecular features of tumor cells such that the tumor cells remember. They have this molecular memory, and they keep on growing, and they undergo metastatic progression. So this biosis can, of course, be as we can achieve this biosis with the result of all these different factors, ranging from drugs to tobacco, and with various other processes, this can impact metastasis, and we can still achieve eubiosis and restore microbiota. So there is a lot of opportunity to use the omics to actually understand the whole process and understand cancer and in a very, very holistic approach, I would say. And this, our aim is to get to a level where we can actually have personalized medicine by understanding all the risk factors, including microbiota and the metabolome changes, and leading to the genomic changes. So there are a lot of challenges ahead, of course, because microbiota interact with various risk factors, and there are multiple confounders. So we do have to understand it much better to get to a point where we know the disease at a better level. So this is again my lab pictures, and I think thank you so much, for your attention.
Wonderful talk, and a lot of work, and it has been really wonderful. We have time for quick 1 or 2 questions. You can either directly call, raise your hand, or put it in the chat box. If we can have quick questions, please.
Okay, so we have a, shall I read the question?
Yeah.
Hi, Deepali. Manisha
Hi.
this side. How are you?
Hey, Manisha.
Hi,
I'm fine,
how are
thanks.
you? It's so nice to see you again here. I feel so overwhelmed seeing you every time being batchmates. So it is so nice to see you. And I took a holiday. I took a leave today to just to listen to you, you know.
Thanks,
So
Manisha.
there is one question, Dipali. Majority of people are diagnosed with breast cancer, even if there is no predisposition or any genetic factor. There are many females these days, especially, who undergo menopause at an early age, and the doctors they opt for HRT, hormone replacement therapy, and when they see that the risks they outweigh the risk of giving HRTs, they outweigh the factors which are that are better, that they that the patients are benefited with the treatment of HRT. So how do we take this? Should the patients undergo HRT, or should they because early menopause means less of estrogen, and there are many other complications being like cardiovascular diseases and so many other problems. So what should they be given, or does this HRT change the microbiota and metabolome? What can you suggest?
Right. So I can say this. So HRT actually is going out of favor now at most institutes. Yes, there are other complications that are associated with early menopause, but instead of HRT, people are now trying to use alternative medicine and maybe using phytoestrogen from soya or from other, like here, tofu is very popular. That is a phytoestrogen. So by taking these alternative approaches, people are trying to avoid HRT, because HRT is associated with multiple other risk factors, I would say.
Mm-hmm. Okay, so what are the alternative therapies would you suggest? Like only these, you were telling, these phytochemicals and all. So every woman
so I was talking about these phytoestrogens that are they're an interesting way, and of course it depends on the symptoms that someone is experiencing, because some people might experience anxiety, some people might have I'm trying to remember, like hot flashes because of the menopause. So there are various aspects of the risk factor of the, I would say, the symptoms, and they would have to be tackled individually with different approaches. So phytoestrogen is just one dietary approach by which some of the side effects can be managed, but then there are others also. But I would say that would depend on each side effect that someone is particularly experiencing.
Okay,
and
thank
Anu
you.
is asking, hey Anu, so Dr. Anupam Varshane is asking.
Hello, Deepali.
Can we have a quick question, last question, because our next speaker is here.
Yeah, right.
So
I'll just
question.
I'll quickly answer the Anu's
question in the chat box.
Right. So you asked about the chemical body washes, soaps, deodorants, etc. So that will definitely have an impact of skin microbiota. And but how does skin microbiota impact other, say, internal organs? That is currently not really clear. But at the same time, I can definitely say that, say, for example, for breast and for any other, prostate, pancreas, those microbiota are inherently not going to change with the external use of these products. So that will most likely be changed by our dietary habits and our exposure to antibiotics, environmental toxins, etc.
The nutrition and all that these days. they are
Right,
talking
nutrition and lack
yoga,
of lack of exercise
nutrition,
and
so
all.
many things everybody is talking about.
Thank
Great.
you.
So because of the paucity of time, maybe Dr. Dipali will be happy to answer questions at the end of the session, or you can email her your queries. Thank you once again, Dr. Dipali, for logging
Thank you
in.
so much.
Wonderful talk. It was really a treat to the ears and mind. Thank you very much.
Thank you so much. I'm putting my email in the chat, so feel free to email me your questions.
Okay, wonderful. So I hand over the mic to my colleague, Dr. Manoj Kashyap, for introducing the next speaker.
Thank you so much, Professor Saluja. First of all, again, very good morning to all. And our next speaker is Professor Achilles Pandey, who is a well-known figure in the field of omics, in particular the proteomics. Professor Pandey did his MBBS from Armed Forces Medical College, Pune, and residency in pathology from the Harvard Medical School. He finished his PhD under the supervision of Professor Vishwan Dixit from University of Michigan, and a postdoctoral fellowship in the laboratory of Dr. Harvey Lawrence from Whitehead, MIT. He had been a visiting scientist in the group of Professor Mathesman at the University of Southern Denmark, and also served as a professor for 16 years at Johns Hopkins School of Medicine in the Department of Biological Chemistry, Oncology and Pathology. Currently, he is a professor at Mayo Clinic, Rochester, and he had been a founder director of Institute of Bioinformatics, Whitefield, Bangalore. Dr. Pandey is going to talk on multi-omics analysis from therapeutic targets and biomarkers in cancer to predicting COVID severity. Over to you, Professor Pandey.
I would like to share my screen. Thank you for the introduction. I think someone will have to stop sharing.
Yeah, Doctor, I think the department has to stop sharing, then Dr. Pandey will be able to upload his.
Okay, we have stopped sharing.
Yes,
Okay,
thank you.
great. Now you can see. Yes, we can see your slides now, sir.
Okay, yes, thank you for the invitation again for me to speak. And I will talk about some of the evolving stories in our group. And the common theme is multi-omics analysis. And initially I was going to talk about COVID-19, but I realized that the major focus is cancer. So I'll tell you about some of the unpublished studies on multi-omics analysis in cholangiocarcinoma as a representative cancer, and how we are using a similar approach for predicting COVID-19 severity. So the goal in cholangiocarcinoma is to identify new kinases as therapeutic targets, and to discover proteins or glycoproteins as diagnostic biomarkers for early detection. And in COVID-19, to study the host response and to use multi-omics analysis to predict severity in patients. Cholangiocarcinoma, as you probably already know, is a tumor of the bile duct epithelium, which is a very aggressive disease and with uniformly poor outcomes. The major classification here is based on where it is located, because the bile duct resides inside the liver and also comes out. So the locations are intrahepatic, hilar, and distal. And other than what is known from genomic studies, that is FGFR fusions, which has provided one potentially attractive target, there is no other targeted therapy for cholangiocarcinoma. And there is also no good biomarker for early detection. CA19-9 is an antibody that has been around for a long time and is used to screen for many different GI cancers, although it doesn't have the best sensitivity or the specificity, but it is still okay. And our vision is that we can find better markers than what is used today. And this is again restating that our goal is to find new targets, and the category that we are mainly focusing on is kinases. So this is the big picture. I won't tell you about all of the different things that we are doing, but we want to look at tumor tissues, and in the tumor tissues, we have the 3 different types of cholangiocarcinoma. We want to do whole exome sequencing, RNA sequencing. Some of these studies have been done, others are ongoing, and proteomics, including protein level measurement, phosphoprotein level measurement, and glycoprotein level measurement. We are also studying a set of 28 or so PDXs that have been developed here at Mayo Clinic, and they represent, again, the diversity of the tumor samples. And in those samples, we're also doing RNA-seq, whole exome sequencing, and proteomic analysis. We have not started FFP samples. So again, there one can do laser capture microdissection and germline sequencing from PBMCs. We have matched serum samples where we can do cytokine profiling, metabolomics, and proteomics. And in the proteome, which is mass spectrometry based, we are doing protein level measurements and glycoproteins, because there our major goal is to find biomarkers. So from frozen tumor tissues, these are human tumors. We have done global proteome and phosphoproteome. And as you probably already know, that there are serine and threonine phosphorylation sites. These are the more abundant sites. But then phosphotyrosine is a very important post-translation modification. It is, of course, phosphorylation, but from an analytical standpoint, it is less abundant. It's nearly only 1% of all phosphorylation. So one needs special tricks to look at tyrosine phosphorylation. And then we are looking at glycosylation changes in the tumor tissues also. And I'll describe some of these snippets of some of these data. And we are doing proteomic and glycoproteomic analysis of serum. So we are using a multiplexing approach using tandem mass tags. What it allows you to do is to essentially barcode different samples. The current schemes allow you to multiplex up to 16 or 18 samples. And then once you barcode them, you can mix these peptides from these samples and do a detailed examination by first fractionating them and subjecting them to multiple LC-MS runs. So there are multiple runs involved, but the advantage is that at the end of mass spectrometry analysis like this, you can identify nearly 10,000 or so proteins. So here the overall strategy is we have taken, at the bottom, you can see the different sets of TMT labeling multiplexing sets, where we have intrahepatic, hilar, and distal, along with a reference channel, some normal samples, and some other unclassified tumors. And we have done basic RPLC-based fractionation, and these fractionated samples are then analyzed for global proteome, and using enrichment strategies that basically rely on chemical enrichment, using titanium dioxide type matrices to enrich for phosphorylation of mainly serine and threonine, and then phosphotyrosine enriched by anti-phosphotyrosine antibodies, and then size exclusion chromatography to look at the glycoproteome. And I'm going to only give you very high level summary data. So in all, we identified nearly 185,000 peptides from 9,900 proteins. And the key here is that the different tumors are in green and the controls are in red. And you can see pretty much very nice separation, except for 1 or 2 odd ones here. And we are still trying to figure out what those are and why this is. But you can see that there is, even though we can distinguish the tumors from normal samples, there is heterogeneity. There are clusters. One cluster is here where you see some signals in the total proteome level, but not in others. There's one here, there are others here. And again, this is a very zoomed out version. And I just would like to tell you some anecdotes of some exciting things coming out. Now, the best part of some of these types of discovery studies is to find new things and to find things that you did not anticipate. So I'm almost sure that none of you has heard of SLC16A3, not a very exciting nomenclature, but even when I tell you what it is a monocarboxylate transporter 4. I don't think that's very enlightening other than telling you it is one in a series of transporters. But if you just look at this signal, and this is going across nearly 60 or so tumors and versus controls, you can see that there is, even though there is a spread, there is an increase of this protein, even though we don't know what it does from controls. So this is the power of these types of studies. And then again, in a zoomed out version, now I'm showing you normalized intensity, because not all tumors are the same. And at the extreme right, what you see are normal samples. And then on the left, you can see, for example, this one tumor, within the limits of this experiment, it is a sky-high type protein expression. Then you have other really high, but then there are other tumors where you don't have any increase at all. We found other proteins such as gremlin-1 and anterior gradient protein 2. And again, looking at a name like that, the first thing we do is, when we don't understand what the protein is, we hit the literature and see who has found what for these proteins. And it also tells us if we are going on the right track, because of course we are not the first ones in the world to study any of this. And immediately we pick up a paper. This was published in 2020, where in extrahepatic cholangiocarcinoma, someone has already talked about gremlin. So this is one of the many molecules we have picked up, which is very low level in controls and then higher levels in tumor type. So the way I measure our performance here is, one is to perhaps feel good that we're on the right track, but then you feel sad that somebody else has already found it. But if you look at the timestamp, we are just 1 year too late. So often when we do these studies, we are sometimes 1 year too late, sometimes 6 years too late. And the hope is always for the future that as we continue on our paths, we'll be the first ones to show some of these observations and then confirm them and hopefully take them to the next stage of using them as biomarkers of potential therapeutic targets. This is another paper on anterior gradient 2, which sounds like a developmental protein, but others have described it in other types of tumors to be overexpressed. And here they say it's in breast cancer patients. But again, so this one has not been shown in cholangiocarcinoma. Switching gears a little bit to phosphorylation. As I mentioned, 99% of overall phosphorylation is on serines and threonines. And the tyrosine phosphorylation is much more restricted, mainly because there are fewer tyrosine kinases. There are about 100 tyrosine kinases versus 500 to 600 serine and threonine and dual specificity kinases. We enrich them by using positively charged metals like iron or titanium. Phosphate is negatively charged. And from this one series of experiments, we can now identify 39,000 serine threonine phosphorylation sites. And of course, not only do we identify the sites, we also can tell if any of them are upregulated or overexpressed in the tumors versus the controls. And again, you can see a very nice separation based on unsupervised clustering, where the controls and the tumors are nicely separated. But again, you can see that there is not one kinase site that is going all across. And here we are obviously not just looking at kinases, but also their downstream substrates that are phosphorylated. We identify 616 tyrosine phosphorylation sites. And when we do enrichment with phosphotyrosine antibodies, we require much larger amounts of material to be able to get detectable amounts of tyrosine phosphopeptides. And here, the signals are really sparse, and you can see that there are certain events going on in certain tumors, but not in others. And then there are other broader signals that are found in multiple tumors. Just zooming in a little here, I'm going to show you a series of tumors and what the relative signals are for any of these tyrosine phosphorylation sites. For example, PRKC, this is PKC delta, the tyrosine 313 is high in this one tumor and kind of low in others, whereas EGFR tyrosine phosphorylation is high in these two tumors, but basically baseline in others. And we are somewhat surprised when we found a 7-transmembrane receptor called GPRC5A. I'd never heard of it, but sure enough, it's phosphorylated on tyrosines and only in one tumor. And then LIN is a non-receptor tyrosine kinase. We found one site to be highly phosphorylated in one, but not others. So again, back to reading, and we see that this GPRC5 is actually a family of G protein coupled receptors. These are still 4 orphan receptors. And even though they are orphan, in English translation, we don't know much about it, but they have been shown or believed to play important roles in human cancer. And some folks have also proposed GPRC5A. That's where we found this phosphorylation site to be an oncogene in pancreatic cancer. But overall, we still don't know much. And this is a smattering of some articles where others have talked about these proteins, and they are not talking about phosphorylation. We have found inducible phosphorylation, but here they are talking about change in levels of the protein in lung cancer, breast, colorectal, and pancreas. And this is another paper that talks about upregulation of this in pancreatic adenocarcinoma. So again, I think although we have found this in cholangiocarcinoma for the first time, I think these are potentially exciting targets, and we are surely on the right track. Switching over to glycoproteomics to find glycoproteins as potential cancer markers. It is known for a long time that aberrant glycosylation happens in tumors, and many of the currently used proteins which are used for screening, especially in GI cancers, are glycoproteins. So alpha fetoprotein, carcinoembryonic antigen, CEA, CA19-9, all of these are glycoproteins. Prostate-specific antigen, PSA, is also a glycoprotein. So now we have technologies where we can look at thousands or tens of thousands of glycopeptides in a single experiment. It's a good time to go back and try to do all of these studies from scratch, again, in the hopes of finding new markers or better markers than what biomarkers are currently available for early diagnosis. And just this volcano plot is telling these studies are ongoing, but here I'm talking about serum samples. So we have identified 1,274 glycopeptides, which correspond to 177 sites from 129 glycoproteins. Many of them are abundant, others are not so abundant, but then you can see these changes. So essentially, we can sequence the peptide and at least deduce the structure of the glycan, and that's how we call it a glycopeptide. And here we are looking at polymeric immunoglobulin receptor, where we have glycosylation at N. These are N-linked glycopeptides, so asparagine 421. And again, you have a very little spread across normal samples, but then you have elevation in tumors. And this is a schematic of, we actually found 2 peptides from this protein, polymeric immunoglobulin receptor, that were upregulated. And here I'm showing you one of them where you have low levels in controls and you have really high levels in higher, but still higher levels in all different subtypes of cholangiocarcinoma. And when we go back and read, it's been talked about in the biology, perhaps not as a biomarker, but as in the biology of hepatocellular carcinoma. And cholangiocarcinoma, although the cell of origin is different, is still kind of discussed in the context, sometimes in the context of hepatocellular carcinoma. So this has been reported, but not in the exact same situation. And it's also been reported to be overexpressed in pancreatic cancer. And again, in these studies, it was not the glycosylation. We are doing glycosylation studies. So again, I think as we do these global studies, we are coming up with interesting molecules. And this is just a few of them. And we are essentially continuing with our studies and pursuing them. So overall, you can see how one can use unbiased proteomic approaches to discover potential therapeutic targets and potential diagnostic markers. But then this is just the beginning of a series of studies that will have to be done in cell line models and patient-derived xenografts for testing individual molecules as potential therapeutic targets. And obviously for diagnostic markers, especially for early detection, we need even larger scale validation studies. Other things that are ongoing in our lab are to use technologies like single-cell proteomics to try to diagnose from single cells or 10 to 100 cells that you can get from bile duct brushings that are sometimes obtained to get a diagnosis before you go in for a real biopsy. So there's a lot of hope and promise, especially as these proteomic technologies evolve. I'll just switch gears and talk a bit about COVID-19. And this is really a story of how a lot can happen in a short time. Our original mandate was to, when there were supply chain problems, and I'll just refer to this major project we did in passing, where our major task was, because of supply chain issues, people were having difficulties with RT-PCR assays, and we were tasked with developing a mass spectrometry-based test to look at viral antigens. While we were doing that, we had access to a lot of nasopharyngeal swab samples, so we started to do many other things. We also got other samples such as FFP samples, lung samples, and urine samples, and we did these types of studies in almost everything that we could get our hands on. And so here, although cell-based studies have been done to look at what happens in cells, we said the best way to answer this question is to look at the cells that are infected, and the site of entry is the nasopharynx. So we took nasopharyngeal samples for proteomic and phosphoproteomic analysis from RT-PCR confirmed positive cases and negative cases. And on the left is a heat map where you can see that these types of analysis, the data are such that, distinguishing positive and negative is very easy. And in this series of studies, we identified many proteins that are basically downstream of the interferon response, but many others that are interferon independent. So a lot of new proteins that were found to be upregulated that had not been described, surely in the context of SARS-CoV-2, but also in the context of any viral infection. This is a similar plot, but this time we are looking at more than 8,000 phosphorylation sites. So again, we have very few cells in these nasopharyngeal swab samples, and we are doing a deep dive to look at the phosphoproteome globally. We found many expected changes, and many of them are interferon-induced genes again that get phosphorylated, and then they are signaling proteins downstream of interferon. We found many interesting cases and some unexpected cases such as opioid growth factor receptor. I think one telling point was when we compared our data from nasopharyngeal swab of what proteins we found to be changing from other cell line studies that had come out by then, normal human bronchial epithelial cell, which is a valid cell model, but it's only one cell in isolation. We basically have very little overlap of proteins that are changing in our system compared to this other in vitro system where these cells were infected in a BSL-3 setting with SARS-CoV-2, for example. And then phosphoproteomic study, these were the changes found there. And here we are looking at the small number of cells that we can get from nasopharyngeal swabs. And we did find many proteins in common, but then others that we found and others that were found in the cell lines, but not in our study. This is the host response study we published. This is the most sensitive mass spectrometry based assay that has been reported yet for detection of viral antigen. It's 98% sensitive and 100% specific. We have also compared our mass spectrometry based assay and RT-PCR with many other point of care tests like lateral flow assays. And as you can imagine, that even though the lateral flow assays detect viral antigen, they cannot be as good as a much more sensitive method, which also happens to be very specific, like mass spectrometry. But of course, mass spectrometry is much more complicated and not even in the same ballpark as a test that you can do in a physician's office. There was a report that spurred us to look at urine, because a paper in JAMA claimed that they couldn't detect viral RNA in urine samples. So we said, can we detect protein? Perhaps they missed it because RNA is unstable or got degraded. And we were able to detect from urine samples SARS-CoV-2 viral antigen. When some of these variants started to come out, we were also able to detect them, and we have published them. But then another thing that started early on is when the pandemic had just begun, we thought, is there a way we can do a multi-omic analysis and use this combined metabolite and protein information to predict severity of disease in patients at baseline? So when they come into the hospital, we get their blood, can we tell who is going to go on which trajectory? I had imagined that we are never going to succeed in this, but we could probably tell the positives from negatives, but this was a tall order. But in any case, we did study about 1,000 metabolites, 900 lipids, and nearly 1,500 cytokines and proteins. This cytokine and protein assay is one popularized by this company called Olink. And while we're in the midst of our study, the types of studies that were coming out were something like this, where people were reporting 17 controlled samples, 21 mild, 23 severe. They were looking at panels of 48 cytokines and 150 metabolites. So we were more ambitious from the beginning. We had a total of 637 samples, of which 182 were COVID-19 negative samples and 455 positive. So this would be the largest study. And of these, 183 were outpatients and 272 were hospitalized. Of the ones that were hospitalized, 139 were severe, and these are WHO ordinal criteria. And then the English translation is that 133 of them were really critically ill. So hospitalized, you don't get hospitalized for nothing, but within that we have two groups, and people who go on for example, ECMO would be in critical, and those who require ventilation would be critical and not severe. So the exciting point for us was this would be the largest set of samples with the broadest representation of patient groups. But the most important part was that these blood samples were collected either just before admission or at the time of admission, not after. So once people are sick, then so many things are happening in a hospital, it's really hard to even take out those factors. But in any case, by that time, you already know that they are going downhill. We also happen to have 24 individuals where we had samples before and after COVID, because they were being followed as part of other studies here at Mayo Clinic, and that this would be the broadest metabolomic, lipidomic, and cytokine analysis of nearly 1,500 proteins. And we also did glycoproteomic analysis of pre and post-COVID. If you just look at cytokines alone, you can look at the key on the right, controls, outpatients, severe, and critical. And the controls and outpatients are close to each other, and severe and critical are close to each other. And basically, using this PCA plot, we could not distinguish the severe from the critical, but we could tell the severe and critical, the hospitalized ones, from the rest. So that was easy. This is cytokine profiling, and what I'm showing at the top is this textbook description is given for cytokines, storm cytokines. The main one is IL-6, then there are others, IL-18, IL-10, and others. And you can see from left to right that, yes, they are going up in severe and critical. Some of the outpatients have a higher level. Below that is a list of cytokines that we discovered as part of our studies. And many of them are going up, and there are some of them, for example, TNF superfamily 11 and 10, they're actually higher in controls and outpatients and going down. So again, interesting biology yet to be discovered. This panel shows what happens in the 4 groups. These are controls. Outpatients don't show much of an increase, but then the hospitalized ones, you already have a significant bump. And in some, you have a trend. In others, like IL-10, although they've been popularized, there's not much of a difference. And there's a small difference in others. So again, it depends on which cytokine you're looking at, and these are the predictors, or presumed to be predictors. Here, I won't go through any of them really, except to show you that we found not just a few, but quite a number of cytokines, growth factors, proteases that are shed into blood that are showing this trend of essentially going up in both severe and critical cases, and in some cases continue to be higher in critical than in severe, or going in the other direction. This is looking at glycoproteome of only the 24 individuals. In this case, we had enough sample from only 21, where we have when they were not sick to becoming sick. And now these are matched samples, and you can see that we have a nice profile of many glycopeptides that are changing. And this is showing you at a site level, beta-2 glycoprotein 1. This is the entire glycoprotein. But then here you got this one site with this structure and the same site with a different sugar. And you are seeing that there is a significant decrease in both severe and critical patients. We also use machine learning. Now that we have so much data from controls and outpatients and those who got really sick versus who were sick but not that sick, and we compared the predictive accuracy of this multi-omic signature of 102 analytes to IL-6, which is the prototypic cytokine in cytokine storm, to the entire cytokine panel. And this is our multi-omic panel. So we were able to predict with far greater accuracy than any of the known signatures when we started. So again, it shows the value that a number of proteins beyond the well-established cytokine storm cytokines can be discovered through these approaches. And we can derive multi-omics, we can use multi-omics analysis to distinguish positive patients from outpatients or those with severe disease. But really, machine learning gives us an additional boost to be able to narrow down to a smaller multi-omic analyzed signature. And it's still not too small, 102. Some of them are only a small set of these 102 are members of the cytokine storm. All the others are new type of molecules, not really connected to COVID-19. And it was a team effort. And I would like to thank all of these members of the team. Roy Smoot is the surgeon who works with us on cholangiocarcinoma. Thank you.
Thank you, Dr. Saab. It was a wonderful talk, very interesting. And this COVID data was really exciting. In such a short time, you have deciphered so many proteins. Thank you very much for the talk.
Thank you.
Can I ask a question? Yes, please go ahead.
Anybody has questions, please?
Yes, can I ask a question? Yogender Singh. Great talk, Dr. Pandey. Question is really different, but probably relevant. So we have seen that post-COVID effect happens in different tissues. It may not be a topic of no research due to the number of receptors, or because the entry point is only mouth or nose. So what could be the reason of this? Like some people get heart problems, some people will get knee problems, some people are getting diabetes. So can you tell me what could be the reason? Possible reason.
Yes, I think it's a good point. Of course, I think the receptors are in multiple places, and people are still arguing about why a given cell type or tissues or organs, whether the virus can enter or not. we have looked at and published, I didn't talk about that study, about the effects of the virus on kidney. This was from some autopsy cases that we got. But I think I would say in the large majority of cases, a lot of the damage is done by these cytokines and the host response, and not directly by the virus. And that's why I think it is really more important to figure out what are all these proteins that are being changed by the virus, because our body is making them, and the virus is just hijacking our own immune system. So if you look at many of the cytokines that we found to be either upregulated or downregulated, they really cover the gamut. And that's why we wanted to get away from these textbook cytokines, because we know what they do, but there is, and this is when we started, what you're talking about, all these multi-organ type dysfunction in COVID-19 was not predicted. So I think, again, going back to these types of discovery approaches, there is a lot more to learn. And I think it is still too early to even make very solid predictions as to exactly what are all the mechanisms that are operative. And in the context of long haul COVID, I think people are now really starting to tool up and now look at the longer term consequences. You were perhaps talking about a little more acute, but again, long term is going to be even more of a nightmare to try to figure out what is happening. But again, I think we need to persist and we need to be doing these studies.
So we have one more question here.
Yes. There is one question. It's regarding Liprogram. Can you please discuss the probable reason behind changes in lipid profile, and is there any relationship between increased level of cytokines on lipid metabolism?
I could only hear part of the question, so could you discuss changes in?
The changes in lipid profile, and is there any relationship between increased level of cytokines and lipid metabolism?
And changes in metabolism?
Lipid profile, and
Yes.
does it have effect on the cytokines and lipid metabolism? Is there any correlation?
I see. Yes, many of them are multifunctional cytokines that have effect on adipocytes and liver and blood vessels. So I think, I don't think I could tell you, even though we found profound lipid changes, I would not be in a position to say that we have found the linear path to saying that these cytokines are doing this. But again, I think this is where making such data available to the community will be very important, because we need many, many thinkers. And even though I think we have a small team and we can think about some, but I think for many of the factors that we have identified to be dysregulated, or even a lot of reading will not educate us as much as the experts know already.
There is one more question from our student, and she's asking, CDC wants to you to elaborate a bit more on the association of increased cytokines in cancer and COVID-19 state.
Increased cytokines in cancer and COVID state. Is the question to connect the two?
Yes, basically in the context of cancer, how it can make things work.
So unfortunately, no, we have not studied that. So the cancer samples were pre-COVID, so there was no COVID connection. And at least even though some of the patients that we had in the COVID cohort, they surely had cancer, but we were not looking at that as a variable. We had cancer in our controls. We had cancer in those who were hospitalized, including those who were severe and critical. So all we can say is it did not play a major role in to introduce a bias in the cytokines. But we did not look for any direct connection between the cytokines in the COVID scenario and cancer.
I think because we already are running the session little late, maybe we can hold the rest of the questions after the session, or people can write an email to Dr. Pandey, and I'm sure he will be happy to answer those questions.
Thank you.
Thank you, Dr.
Thank you, Manoj. Yeah.
Yes,
Thank
Pandey,
you,
for such a wonderful
Dr. Saluja.
talk.
Yeah.
it was fantastic
So
talk.
we-
Thank you so much for explaining the time. Thank you. And
So we move on to the next speaker of today, session 1, Dr. Shantanu Sen Gupta. Dr. Shantanu did his graduation from University of Calcutta, West Bengal, and then PhD from Chittaranjan National Cancer Institute, West Bengal, in cancer biology. He then took a fellowship and went to otolaryngology head and neck surgery department at Johns Hopkins School of Medicine, Baltimore. And currently he is assistant professor of pathology at Michelle Cancer Institute, Alabama, USA. And his major area of research and the team that he is in Mitochondrial Research Laboratory, Department of Pathology, College of Medicine, Department of Biochemistry and Molecular Biology, Cancer Biology Program. His thrust area of interest is basic and translational cancer research, genomics, epigenomics, genetic alterations, and metabolic programming. And they are interested in to study the early detection, monitoring of biomarkers for racial health disparities, genome and epigenomic characterization of cervical, ovarian, head and neck cancer, pancreatic, and prostate cancers, and so on. So their main area of focus is understanding the role of mitochondrial genetic alterations and metabolomic reprogramming in cancer health disparities. And then they are trying to develop mitochondria and circulating extracellular vesicles-based biomarkers for early cancer detection, monitoring, risk assessment, and then which could be used as a therapeutic guidance. With this short brief, I welcome you, Dr. Shantanu, and I'm looking forward to your talk on mitochondrial dysfunction. Very interesting, and his title of his talk is Mitochondrial Journey of Human Cancer. Welcome, Dr. Shantanu, and floor is yours.
Yeah, can you hear me clearly?
Yes, we can hear.
And can you see my slides also?
If the organizers, yes, we can see
Yes.
your. No.
Okay, thank you for your kind introduction, Dr. Saluja. And I would like to thank the organizer for inviting me to share my work with you. And I'm really like inspired by the wonderful talk by the preceding scientist on Dr. Sharma and Dr. Pandey. Very impressive work that they have shared with us. And with this, I'm going to share my work with you today on mitochondria in cancer. I'm unable to move my slides. Oh, okay. So I will start with my studies in lung cancer, and briefly we'll talk about bladder and other cancer type as well. So as we all know that globally, lung cancer kills more than 1 million people worldwide and over 130,000 in United States. And it is very important to understand the key genetic alterations that drive tumorigenesis, and so that we can develop useful biomarker for early diagnosis, prognosis, or therapeutics, because still in lung cancer, early diagnosis is a big problem and therapeutics are also very limited. So as our previous speakers have given a compendium of their thoughts that how multi-omics approaches can be useful to develop useful biomarkers or therapeutics for different cancer types. So as we all know that this is an era of multi-omics approaches with deep sequencing. And if you look at the TCGA data on lung cancer, you would appreciate that thousands of patients have been sequenced already, and this is a list of 20 top mutated genes on lung cancer. This is from the COSMIC database, and you can see EGFR, KRAS, BRAF, and other genes which are very good target for therapeutics. So in this era, we can utilize this information for develop not only markers, but also novel therapeutics. So with that information, so I will talk about briefly what are the different types of lung cancer. So lung cancer basically are of two types, small cell and non-small cell. And non-small cell type is the most common type, and which has three different types of histology, basically adenocarcinomas, squamous cell carcinomas, and large cell carcinomas. And adenos and squamous are the most common histological subtypes, but sometimes you may also see adeno and squamous being mixed, and this which offers clinical challenges for therapeutics. And in terms of molecular pathology, along with the histology, we use epidermal growth factor receptor EGFR, ALK email translocation as a marker for detection. And treatments option, we have surgery, radiation, chemotherapy, which are the conventional mode of therapeutics. And as I mentioned earlier, with the cutting edge technologies with deep sequencing, we can now have more targets in addition to EGFR. We have ALK, ROS1, BRAF, NRTK1 inhibitors are in place, and of course, PDL1 and PD1. But unfortunately for lung cancer patient, particularly the patient who have mutation for EGFR, these therapies are not very effective. So I'll briefly show the molecular pathway that are very well established in lung cancer. The classic EGFR pathway, which is very common among the non-smoker, and the KRAS pathway, which is common among the smoker patient. So I must tell you at this point that adenocarcinoma histology subtypes are very common among the patients who are actually non-smoker, as opposed to the squamous cell carcinoma histology is very common among the patients who are basically smoker and bear KRAS mutation. So as I mentioned earlier, there are many lines of therapy targeting these molecules which have been discovered over the years by next-gen sequencing. Many inhibitors are in place that can be used for treating lung cancer patient. And this slide shows the different complicated histology that the pathologists encounter while making a treatment decisions. And the good thing about multiomics that we can keep identifying the molecular alterations at different time points when a tumor progresses from normal to pre-neoplasia to neoplasia. And with this technology, when you identify the key genes, and these genes along with the histopathologic finding can be used for molecular diagnostics, which are the tremendous advancement, I would say, in the era of multiomics. So for my project, I was interested in analyzing some primary lung tumor and some follow-up biopsy specimen from lung cancer patient through a multiomics approach. So what I did for the first set, so we have, for example, like 5 patients, and we have the primary tumor from all the patients. And these patients came into the Johns Hopkins Clinic for follow-up after their primary removal of the primary tumor. And they were followed up through a process called autofluorescence bronchoscopy, which normally can detect any abnormal lesions following the first surgery among the patients. So this is some data which was done in Hopkins long back by Dr. X Young. So we have these class 3 lesions which are suspicious for mild, moderate, or severe dysplasia. So we have these lesions in our hand. So what we did, we did histopathology, and we can see that either you get dysplasia or carcinoma in situ, or get full-blown carcinoma in the follow-up biopsy. So we have the primary tumor and the follow-up biopsies of these patients. So now the question is, why would you do mitochondria genome sequencing? How it is going to be beneficial for your work? So the reason being, mitochondrial DNA mutation occurs in different cancer. They have been reported, I think, almost in all solid tumor types. And it's very easier to detect because of the high copy number in cancer cell. A single cell in our body has around 50 to 100 of mitochondria, and each mitochondrion, they contain 10 to 20 copies of the genome. So it is very easier to detect. And we have shown earlier that mitochondrial DNA mutation functionally contribute to cancer progression. So, and they have also been implicated for therapeutic and biomarker development. And you will see many studies are coming up where now scientists are targeting respiratory complex 1, 2, or 3 for cure cancer. So in a nutshell, this is the structure of the mitochondrial genome, which is basically a 16.5 kb genome, which is a double stranded circular molecule. And it encodes for 37 genes, and of which 13 are protein encoded. That means there are 5 respiratory complexes through which mitochondria makes energy, and 7 genes from respiratory complex 1, 1 from complex 3, 3 from complex 4, and 2 from complex 5 are being made by the mitochondria themselves. And amazingly, all the other respiratory complex protein are nuclear transcribed. They're translated into the cytoplasm, and they have mitochondria has a unique import machinery through which these protein are being imported into the mitochondria. They get assembled with the mitochondrial protein and turn on the OXPHOS system. So another interesting thing about mitochondria is the generation of heteroplasmy. So I'm going to talk about the heteroplasmy thing. So at birth, mitochondrial DNA has the wild type DNA. So there is no mutation. They're happy, but with environmental and genotoxic damages, the mutation can occur in the mitochondrial genome. So now you have a state where you have both wild type and the mutant mitochondrial DNA. Since mitochondria do not have protective histones, they are more prone to have mutations. So you have a situation where you have a mixture of mutant and wild type mitochondrial DNA with more heteroplasmy. Either you can achieve a state of homoplasmy with mutant mitochondrial DNA, which is now been seen in cancer. With the advent of deep sequencing, we can identify if there are mitochondria sitting with selective mitochondrial DNA mutation. So with this background, what we did, from the 5 patients, we have the matched primary cancer and at least 5 mucosal biopsies from the same patients, and we have normal lymphocyte to compare with. So we have isolated DNA, genomic DNA from all the cases, and we then amplify mitochondrial genomic DNA and do a next-gen sequencing. And so I'm going to show you some data from a couple of patients. So this is a patient who had a tumor on both the lung, and we have primary tumors and 5 mucosal biopsy lesions from the same patient, and we perform mitochondrial DNA sequencing. What we have found is, which was very surprising, that many of the mutations are shared between the primary tumor and the follow-up mucosal biopsy, which were taken after the resection of the primary tumor, which indicates that there are some clonal relationship between the mutations of the secondary lesions and the primary tumor. So that gives you an idea that some mutations may be necessary for the tumor to survive or to develop a second primary lung tumor, which is a very common problem in lung cancer. The patients are always at a risk of developing a second primary tumor. So this is another patient where we have shown that many mutations were shared between the primary tumor and the developing secondary lesions. And the mitochondrial DNA content was found to be an indicator of aggressive tumor. And we have also measured mitochondrial DNA content in this patient. And we have seen that compared to the normal, the non-tumorigenic lesions and also the tumor have an increased content of mitochondria in all these patients. So apart from that study, another multi-center study that we have conducted, and it was conducted in collaboration with UT Southwestern, and also Mayo Clinic with Dr. Jean Jain. So what we have in this cohort, we have 60 patients, all lung cancer patients. We have 30 patients who were never smoker, and we had 30 patients who were smoker. So what we did EGFR KRAS sequencing, which was done in UT Southwestern in Adi Gazdar labs, and we have done the mitochondrial genome sequencing, and then kind of integrated all the information to see what is going on with these patients. So what was then we found a lot of DNA mutation in these patients, and which we have noticed that the patients who have the non-smoker patient who have more EGFR mutation tend to have more mitochondrial DNA mutation too. But that was not the case in the patients where we have KRAS mutations. So basically, we found a correlation between increased EGFR and mtDNA mutation. And we have also compared between smoker and non-smoker patient in terms of DNA mutations. And we are expecting that we will obtain more mtDNA mutations among the smoker. But that was not the case. We found that number of mutations were much higher among the never smoker compared to the smoker patient. And we also have checked the racial distribution. We have patients from Asia and Caucasian. We have seen that Caucasian patients have more, Asian patients have more mutation compared to the Caucasian patient. We also measured the mtDNA content in this patient, and we found that the never smoker patient have more mtDNA content compared to the compared to the smoker. So apart from that study, we are also interested to know. So we have done mitochondrial genome sequencing in solid tissues, but in order to be able to develop non-invasive biomarker, can we detect them in body fluid samples? So we had a project in bladder cancer where we have a primary tumor, normal tissues, and we have urine from each patient. And if you look at the table, many of the DNA mutation that we've seen in the primary tumor were readily detectable in the urine. So that tells you that mtDNA mutation which are associated with the tumor can be detectable in urine and maybe any other body fluid samples. So this is very encouraging from the biomarker point of view that this information can be useful. So, but the obvious question comes into everybody's mind that, okay, you have done some sequencing and it is obvious you're going to get some mutations, but do they have any functional relevance in tumorigenesis? So because this mutation could be just simply passenger mutation, can they drive? So as I mentioned earlier I have shown that said in bladder cancer that mtDNA mutation can promote tumorigenesis. So I'm doing a study in prostate cancer where in some African American patient we have seen a particular mutation from respiratory complex 1, which is encoded by the mitochondrial genome is very high. So we sought to clone that mutation and make stable clones in LANCAP prostate cancer cells. And we make sure the protein the mutant protein is in mitochondria. Then we measured mitochondrial ROS, which appears to be very highly expressed in the cells expressing the mutant protein, and cells are consuming more oxygen and making more ATPs. So that means this mutation may have a role in promoting tumor by enhancing the OXPO function. So currently I'm just studying the mechanism exactly how these mutations are having this effect on the cells. And we also measured growth and invasion potential of the mutant cells harboring that mtDNA mutations. And we are seeing some encouraging data. But again, this is unpublished, and I've just submitted a grant using this data, and I'm just looking in depth into the mechanism if there are any specific role of this mutation in promoting tumorigenesis. So that is one part of the multi-omic study. So the same DNA what we did also, We also performed a whole genome SNP array using the same DNA from the patient to look at what are the other nuclear encoded genes that are being altered. So for that, we have taken 250 SNP array, which is a very, very powerful tool. So basically in this method, you use hundreds of thousands of SNPs, which are intragenic. And by measuring this SNP, one would be able to tell you whether a certain genes are lost or amplified. So with this approach, we are able to identify some genes for which we have a lot of loss of SNPs. So that means these are basically could be potential tumor suppressor genes. So from that gene, so we need to validate whether your high throughput data is valid and you can reproduce them. So I have picked a one gene, which is known as endophyllin A1 or SSTGL2. So which has a role in clathrin-coated endocytic vesicle receptor, vesicle trafficking. And another important aspect that these genes also interact with the tyrosine kinase receptor, including EGFR. So it indicates there is a severe loss of this gene. So what we did, we, in collaboration with Dr. Jean Janet, she used to be in Mayo Clinic where Dr. Pandey is now. Now she's in a company. So we allotype like 116 lung cancer patient sample with using marker from this from within these genes. And we can see clearly allelic loss of this gene in more than 50% of sample. We also did FISH, use probe from 9p21, where SSTGL2 is located, and also use a control probe from 9q, and you can see a loss of one copy of the gene. And also we checked at protein level, and we can see significant loss of this protein in lung tumors compared to normal. And when we looked at the TCGA data, it corroborates well with our finding. And RLA level, so this gene appeared to be severely downregulated in lung cancer and could be a good biomarker. So basically what it tells you, these tools are very powerful, and if you can validate, it is reproducible and it's very exciting. So another interesting finding we made from one of these patients, we found a homozygous deletion. And you can see, so this is one patient where you can see the tumor and non-tumorigenic lesions that were taken out from the patient when they came from follow-up, and normal lymph node tissues were act as control. And you can see this is the area of genomic amplification in different region. The beauty of this technique is that you can identify not only the genes that are being amplified, you can identify the specific exons the mutation is located, and also you can identify potential microRNA binding sites. And here is the location of the homozygous deletion that we have seen. Now the question is, can you identify the genes that are located within this deleted region? The answer is yes. So we identified a bunch of genes within this homozygous deletion. So I'm working on some of these genes now in the lab. And this is other than this molecule, I was also interested to look at through the SNP whether I could see some proteins which function in mitochondria are also altered. As I mentioned earlier, mitochondria host for around 1,500 proteins for different function like OXPOS, TCase, amino acid metabolism, DAM signaling, calcium signaling. But amazingly, except for the 13 proteins, all are nuclear transcribed. This is amazing. So when I looked at the other mitochondrial protein, I found a 40 gene signature, which were either their copy number was lost or their copy number was increased in this in the same tumor. So in the same patients, in the lesions, we can see it changes in the mitochondrial DNA and changes in so many genes. It's so overwhelming. But you can orchestrate and coordinate all this data to find out different ways to develop markers. That is the whole point behind this integrative genomic studies. So then I looked into a TCGA database with my 40 gene signature and try to, and we have a lot of patients, and I found out that of the 40 genes, 35 genes were found to be overexpressed in lung cancer tissues compared to the normal. So, and I'm validating some of them and doing some functional studies with this data. So to summarize the work, integrative multiomics approaches can be very, very powerful, and they can aid to develop cost-effective diagnostic and prognostic biomarkers, and you can also even develop targeted therapies based on this outcome. And mitochondria is less well studied. I must say, like TCGA kind of ignored mitochondrial genome, but I'm on it for some long time now and trying to utilize this information to develop markers. And this cutting edge integrative genomic screening can be very helpful for to the pathologists for molecular diagnostics, so that better treatment choices can be made. And I would like to thank Dr. David Sedzansky, Rex Young, and Bill Westra from Johns Hopkins University for giving the opportunity to work with them. And I would like to thank Dr. Herrera, our chair, University of South Alabama. Dr. Rajay Singh and Seema Singh are my collaborators. Dr. Carter is my team pathologist. Dr. Pierce is my clinical coordinator and collaborator in the gynecologic cancer that I'm working with in cervix and ovary. Casey Daniel is a social scientist and epidemiologist who helped me on social epigenomic studies. Dr. Karan Singh from UT Health is my team biostatistician. Dr. Galies is my consultant in lung cancer studies. And I would like to thank all the patients for kindly providing their samples, without which you cannot do anything. And thank you all for your patient hearing. And I will stop here. And if you have any questions, feel free to. Thank you.
Thank you, Dr. Shantanu Dasgupta, for a wonderful talk. And it was really very intriguing to see how mitochondria can be so much important. We always keep thinking about it's mainly into apoptosis and when we are talking about and all those things. But yes, so gradually I have just a quick, so gradually you think all the mitochondria DNA get mutated, and they kind of these mutated mitochondria genome replace all the mitochondria in the cell, and that is why the severity keeps on increasing.
Yeah, so with the advent of like with the advent of deep sequencing, we are starting seeing like more and more copies of mutated DNA in the mitochondria. We use now droplet digital PCR and next-gen sequencing. But normally in a normal cell, if you have too many mutations, the apoptosis will turn on, the DAM signaling will turn on.
Yes.
But what happened in cancer cells, they are very clever. They block the DAM signaling, apoptotic signaling pathway, so that mitochondrial nuclear retrograde signaling does not occur. So they are very clever. They have a way to think how to
bypassing that.
Yes, that is very fascinating. So DAM signaling is very well known. Yeah, under normal condition, if the mitochondrial damage, they are unable to repair by fusion or fission. This signal will go to the nucleus to so that intrinsic apoptosis can turn on, but it does not happen in cancer cells.
Yes. Any questions from the audience? Sir, if we can, you can stop sharing your slides and we can have the. Any questions from audience? So, if there are none, I may ask another question.
Dr. Saluja, there is a question from audience.
Yes,
okay.
Please.
Hello, Dr. Dasgupta. That was very nice talk. I just have a question about correlation of mitochondrial copy number with cancer. Is there any correlation or any specific relation to their copy number with cancer? And can you just tell me?
Yes. So in prostate and breast cancer, I remember, and also in lung, increase in copy number have been associated with aggressive form of the development. In prostate, for sure, the DNA copy number is high, and when there is high, the patient, they found that in aggressive patient who are diagnosed with advanced stage tumor, they have more copy number of the mitochondrial genome.
Okay, so we can say that it can be used as prognostic factor.
Yes, of course. But for that, you need to do multi-center study with a large number of patients, because when you are talking about patient, you must have a lot of patients.
Okay. Thank you very much.
Big cohort is required.
Yes, one more question, please.
There is one more question.
Sure.
Yeah, can you stop sharing your slides, Dr. Saab?
Yeah, I did.
So then we come to the screen.
Dr.
Questions, please.
Yeah. Am I audible? Hello? Yeah.
Yes,
Hello?
you are audible.
Yes, Dr. Dasgupta, thanks a lot for this wonderful job. You mentioned about some of the genes promoting oxygen consumption, some of the mitochondrial DNA,
Yeah.
right? And hence promoting tumor growth. Maybe I missed, could you just highlight more upon that? What exactly they are doing, the mechanism?
Yeah, that's what we are studying. Like when we introduce the mutation in the cells started producing a lot of ROS. And under normal conditions, if you have a lot of ROS, cells die, right? But cancer cells do not die in high ROS. I'm just overwhelmed myself. I do handwork in the lab. They do a lot of ROS, they don't die. And ROS has the ability to turn on a lot of oncogenic pathway. For example, NF-kappa B pathway, CXCR pathway. So in that case, we have seen that CXCR4 pathway is upregulated. So that is the possible mechanism. But what I'm working on, that the basic question is how the mutant protein is actually causing the induction of us. Is it through electron leaking or any other known mechanism that I'm just delving digging into the detail? Because if we can figure it out, then it is something interesting. Does that answer your question?
Yeah, indeed. I would look forward to your further research. Yeah, sure.
Okay, thank you.
I'm sure there are many more questions, but because of the time, and it must be midnight for you guys.
Yeah, that's okay. We keep writing grant in midnights, so don't worry about it.
So thank you very much on behalf of the organizing committee and on my own behalf and audience. We would like to thank all the three speakers of today's session 1. Who have really spared their time, and that too late night in USA, and to enlighten us with some of their recent work, unpublished work. Thank you once again, and over to the organizers for the next program.
Thank you all. Thank you.
Good night.
Take care. Bye-bye.
Thanks to all the speakers. Thank you so much for making it a wonderful session. Thank you so much. And thank you, Professor Saluja, also for sharing the session. Thank you.
With this, our first academic session gets completed. I would now like to request Professor Ashok Kumar Singh to give away the token of appreciation to Dr. Manoj Kashyap.
Madam, your token of appreciation is still due, and you have to come to the department.
Yes, I will come to the department. No issues. I will be coming.
Yeah, so we wanted to see you physically, but it's okay. I think it was quite early in the morning. We could not actually. We missed you.
Yeah, no. So I was not sure how it is planned. So and being the first session, little bit of ignorance also. I will be joining the other sessions physically. I will be coming in between. It was a wonderful session.
Yeah, it was nice. Thanks again to all the teachers.
We now request you all for a short tea break. We will again assemble at 11:15 for inaugural function.
11:15.
एक्स्ट्रा ये ना और कल लाके आप 30 बोला ये रख लो।
25 आ गया।
ठीक है, आप देख लेना हिसाब
के सामने वाले
ये पीछे चले गए। 20% कैसे आज का सबसे सुनिए